Okay, so my name is Doreen. I am a data manager in a developmental psychology lab in Leiden. And I was hired to help develop a vision on open science for our lab. And also see how we can implement open science practices in our lab. And while doing that, I also bumped into the issue of sharing MRI data, because we collect a lot of MRI data in our lab. And in the previous hackathon, we also notified that many researchers don't really know where to start when they want to share MRI data, just as I didn't know where to start as well when I started working there. So that's why we came up with the idea to make a decision tree for researchers that could help them decide what they could do in which stage of their research. We have already started a decision tree, but it's only for Leiden and it's also not finished. So I also wanted to make this decision tree also applicable to more universities and maybe even the EU, if that's possible. So that's what I wanted to do in this hackathon. So the goal is to draft a decision tree for sharing human MRI data. And the sub-goals, they are to formulate more questions or maybe more relevant questions and prioritize them a bit more. And also come up with solutions that are applicable to other universities, because mostly our solutions now are mostly focused on Leiden University, and I think they should be broader. And of course finish the first version, and also maybe look at how we can implement this tree, so how can we reach other researchers who are also struggling with this problem, and maybe also just practically where can we host it and how can we make it, stuff like that. If you want to help, you can help in all kinds of ways. So mostly you can think of which questions does the tree need to address, which regulation should be included besides the regulations that we already have now. And also use cases. There are no use cases yet in the decision tree now, examples. So I think they should be included as well, for clarity. And maybe add some resources that we haven't included yet, or find other repositories that we can use. And as I said, how can we host and disseminate the tree and that stuff. Everyone is welcome to join. There's also no maximum number of participants, because I think everyone can contribute if they want. Just if you're interested and you can use Google, then you already qualify. So if you know anything about privacy issues or ethical questions or data sharing in general, or maybe already something about MRI data sharing, you're also very welcome. But in principle, I think I welcome everyone. So if you're interested and you feel like you can help and you want to help, then you're very welcome. Thank you.
Hi, my name is Xenia. So we are all more or less brain scientists, right? So we are going to discover the most complex organ in the body, and we will solve how the brain works pretty soon, right? But aren't you missing like one part, or some parts of the brain in all our analysis? Like in most parcellations and images, no? Well, there are two small organs that are normally neglected. So some people of you might know this meme. So this is, maybe the brainstem and the cerebellum. They are called subcortex by MRI scientists. Well, in neuroanatomy, we didn't call them like something that was below the cortex. We called them, like, they were separate parts of the brain, and they were equal, but now we call them subcortex, and usually we leave them out of the analysis. So what are we going to do about it? So I thought in my hackathon project we could compare cortical and subcortical dynamics in some kind of match, like some kind of boxing range. So to see who wins, and if you like, you could join this project. So there are different skills that might be needed. So even if you are not have not done any programming yet, it could be an interesting project for you. So I would like to research, discuss, and decide which metrics might be most informative for comparing two brain regions, and then calculate brain dynamics using two different sets of regions and compare them. So it's partly researching how to compare those regions, and then also figuring out which toolbox might be able to do this. And so it's a kind of intersection between neuroimaging and computational neuroscience. And those who are interested could also create mind-blowing visualizations as well. So you can talk to me or join the Slack channel, and if you have any questions, if you would like to join, so hope to talk to you soon.
Hi, I am Faruk from Maastricht University. A couple of weeks ago I submitted my PhD thesis, so I feel like I can do anything now. So I decided to do something that I wanted to do for a long time, which is exploding brains. Research questions are, can we explode some brains virtually, of course, safe? And how many different types of explosions we can generate before the visuals get boring? Research answers. So 1, yes, we can explode brains virtually. And 2, more research is needed to determine when it gets boring. So my methodology is having some MRI data, doing tissue classifications, then particle simulations. This is the most fun part. And of course, introspection. Is it enjoyable? Is it boring yet? Like, what can I do? What, where, and how? So I have submitted an issue in the OpenMR Benelux GitHub page. You can see more details there. There are some reading materials that you can see whether you like it or not. And also I'm planning to implement everything in a GitHub repository too, in a completely open source, which is called the slowest particle simulator on Earth, because I am planning to use Python to implement it. That's why. But as long as it works, it's fine. So thank you.
Hi, I'm Johannes. I'm an assistant professor here at the Department of Cognitive Neuroscience. My office is just there. It's going to be really hard to pitch a project after the exploding brain. So what I want to do is a functional parcellation of the mouse brain. And the concept here is depicted using a Mondrian painting. But the general idea is that we are not alone on this planet. There are other species, and some of these species are actually investigated and used in research. And that's the case for the mouse. So on the left-hand side, you can see the human brain and other mammals, and you see that the mouse is quite far from an evolutionary perspective, also has a tiny brain, but it's heavily investigated in neuroscience. And when we study neuroscience, the first question we want to address really is, how are the different parts of the brains organized, how are they clustered, how are they segregated? And people have spent a huge amount of time looking at that, and these are examples such as for the mouse brains, the human brain, and a couple of monkeys. So when we actually do atlasing, there are two different ways, and traditionally it's been done using histological analysis, looking at cell densities, so how much neurons there are and how they're packed, and that's how we got these beautiful yet slightly boring atlases. But when you work with humans, unfortunately, tissue is not as available because somehow humans don't want to part with their brains, at least not when they're alive. And people have been using instead functional MRI as a way to generate parcellations. So they use algorithms on data-driven to generate the atlases. So we wanted to ask the question, can we generate a data-driven mouse brain atlas how does it compare to cytology or histological atlases? So then that way we can actually validate the parcellation algorithms. And then can we also apply it to other modalities? So there's a lot of data on databases for structural connectivity or gene expression, so we could use that. And of course, if we had an unlimited amount of time, we could also compare it across species and get data from rats and monkeys. Where does the data come from? I've shared single-handedly all of my data since I started my PhD, so in when I finished in 2014. So, and I think I've accumulated over 1,000 high-resolution mouse fMRI scans, and they're all available now unprocessed on repositories, but I'm also making them available pre-processed into a standardized space, so we can start making the atlas. And the easiest parcellation algorithm I could find, which we could implement, is the so-called Gordon method, and then that's the kind of steps that you need to do. As far as I understood, it was done at MATLAB for that particular instances, but I think the functions are also available in Python, so people could try different things. The part to look forward for is, of course, the very end when we celebrate. With that, thank you very much.
Hello everyone, I'm Pieter van der Malen from Ghent University, and this is
Robert Oosterveld from the Dons Institute.
Okay, so we will present a project on bits and integration with MATLAB now. For the people who don't know bits, bits is a simple and intuitive way to organize and describe your neuroimaging and behavioral data. Bits is not an image format like DICOM nor NIFTY, but it's more of a formal specification. It has been published in Nature Scientific Data, and it uses a human and computer-readable folder structure and file naming convention, and also you can store metadata using text files, ASCII files in tabular form or in JSON format. But now there are some challenges when we use bits. So we need to interact with our bits data set. So we have a lot of here mouse data sets of 1,000 scans. So we need to be able to read this easily and to write to the bits data set easily. And we also want to query data. For instance, we want from these kinds of subjects, all these kinds of images with this kind of metadata. So we need to be able to easily interact with our data set. Now, the thing is that there are continuously new extensions being added to the bit specification, so it's a continuously evolving specification. And most open source tools are written in Python. Now, we do like Python, no? But we also do like MATLAB, so we really need something to do with MATLAB. And also, the current tools available now can only do one of these. So we can do the validation, and especially the bits validator is a very, very useful tool, but only does validation. We also need to convert our DICOM data or other kinds of data to bits. There are specific tools for this, but only limited to mostly a subset of the bit specification. We also want to read the data, and we want to use the bit data to be processed. And all these tools in Python can only do one or just a subpart of one of these steps. And there's also no bits MATLAB community, so we are the community now. So it's quite limited.
Well, so there are more people working on this, but it's still very immature. And
yeah.
What we plan to do in the next 2 days, 2 or 3 days.
I will give it to you.
Yeah, it's actually to get more of the people that are working with MATLAB and bits dataset, to get them together and to work together and to share more code and to develop a joint code base. There are some tools in MATLAB. There is the bits MATLAB package, but it's still very immature and incomplete. That's what we'll be working on.
Yeah, especially. But it's also important to define what you really want to know from bits. What you want to do with the bits data set. You can, as I'm an engineer myself, so I can think of a lot of things we can do with your bits data set, but these things need to be useful and meaningful. I can implement a lot of functions which are maybe never used. So we need a well-defined package. So we need to define a good interface in bits and MATLAB. Maybe you can also think about a framework or architecture and what's really want to do, what Robert said.
So if you're working with bits and if you're working with MATLAB, then you can consider helping us to work together to making tools that help us all. Yeah?
Indeed. We also have some, maybe the skills you need. You need something about bits. You don't need to be an expert, but if you can connect and talk about bits, then you're mostly welcome. Programming skills are, for the open discussion, not necessary. So we also want to address users of tools like bits readers. So what you want from us as developers to implement in MATLAB, how you want to interact with the data, etc. And we also have some ideas for the coders, like Robert told. If you have some magnificent ideas, you're mostly welcome. So I hope you can attract some people to work with the nerds amongst us. Okay, thank you very much.
So thank you very much. My name is Barbara. I work in Liverpool. I'm very happy to have been accepted to the MR Benelux. England is not a Benelux country. I'm proposing the cortical lesion finder project. And in particular, I've been working on data collected on patients with epilepsy and healthy controls. So the idea for epilepsy is that in many patients, about 40% of them, seizures continue and are not well controlled, either by anti-epileptic drugs or any other treatment options. And surgery also doesn't seem to be a clear option in these patients because no epileptogenic lesion can be found on MRI just by visual inspection. So these cases are discussed in the multidisciplinary team with all the clinicians, neuropsychologists, surgeons. However, the neuroradiologist will review the T1 weighted images and the T2 flare scans to try and find lesions, but frequently they're very subtle and will have to be identified quantitatively. So this is what we have tried so that ultimately the patient could have successful epilepsy surgery. So, and this is what this abnormality looks like. This is a focal cortical dysplasia site. If you compare the left hemisphere to the right, which is intact, you can see that there's a funnel-shaped lesion, and that cortex is very thick in that area. And so the idea is to automatically identify focal cortical dysplasia from these images. So I've implemented the methods in MATLAB, but as we've just heard, MATLAB is not freely available and has some problems when you make it open source as well. So the idea is to transfer the code that I wrote from MATLAB into Python. We basically take the T1 weighted image and normalize and intensity correct it, and then we use the anatomical atlas label mask that is available in SPM to only take the cortex, and then we can check for voxels that have blurred voxel intensities, so they're neither clear gray or white matter signals, so we can make a junction map, and where you see clusters of voxels, well, that's where there's a likely site for focal cortical dysplasia. And we can also use the gray matter segment to look at gray matter thickness. So then we smooth the images, and then obviously we can do a t-test between a single patient and the controls. So I have a control database of 40 subjects that we can compare to. And so then you end up with statistical significant results, and here I've plotted the T2 flare images, so lots of blurring in this hemisphere, and it's picked up by the software. So I also have a GitHub issue, so if I can just, so on the, we've already worked on this at the OHBM hackathon, so we've implemented some steps already. The DICOM to NIFTY conversion and some intensity correction, and I'd be happy to know if you have any skills in MATLAB or Python. Even if you're just a beginner, happy to work with you. So you can contact me on the Cortical Lesion Finder Slack channel. Yeah, and happy to hear if you'd like to work with me. Thanks.
Right, okay. So it's a bit of a lofty title. So with eCovidas, you will never have to write a method section again. So imagine I bake this cake and you love it and you want more of it. And you want to do it yourself. And you ask me for the recipe. And then I tell you, oh, it's very simple. Just mix all the ingredients and put in the oven. You'd go like, that's not really helpful, right? And yet, when we read some of our neuroimaging papers, you go to the method section and you go, pre-processing was done. We sort of realign and slice time the images with SPM. And you go, wait, that's it? And we know that actually just pre-processing has a big effect on what results you're going to get in the end, right? So to sort of fix this problem, OHBM created this committee called the COBIDAS committee, which came up with those reports that are pretty much a set of guidelines, which should be sort of what you should actually put in your methods section and how you should report your results. And the report is super useful, except for the fact that it's pretty much a 60 to 70 pages long document. It has a very useful checklist table at the end, except that most of the time it has plenty of stuff that does not concern you. So, like, you know, if you do fMRI, you don't necessarily want to have to skip to the 3 pages of DTI, right? So what was started at the hackathon at OHBM last year was to create an app where you would just sort of have to click through all the things that you did, sort of type in, and we trying to minimize the amount of typing you actually have to do, sort of present people with like a set of choices and they can just quickly go through this thing. So that we would have a more comprehensive, way to report methods that would be embedded in the end in sort of a small text file, a JSON file. And so we have a prototype app at the moment, which is sort of really bare bone of what we're trying to do, and it's just based on sort of the metadata that you would need to report if you want to upload your fMRI or neuroimaging data in general to NeuroVolt. You can actually check the link to this link to try the app for yourself. The next step that we want to go, or want to have actually like a first sort of full prototype of the app, is to sort of want to make sure that once you've filled in the whole checklist, you would just have to press a button and it will automatically generate the method section for you. The thing we need for that is to just create sort of the text with like the blank spaces where all your choices that you have made are gonna go, right? So we have just a very small goal for this hackathon. I mean, very small. It's not, it could be big, but we want to start small. It's like pretty much a text template that would in the end be used by the app to write the method section for you, right? So the prerequisites for this are just very low. You don't exactly need to, so like there won't be any coding because most likely we're going to start working in a Google Doc and trying to create like template text that we'll be able to use in the end, right? Obviously, if you know about MRI and about some softwares, it would be helpful. But if you just want to actually just drop by and want to learn more about the app and the project, just feel free to just get in touch. And I won't be alone. I think there'll be Cass with me, maybe helping here and there, and Susie as well. So if, just come to us, just drop us a message in the Slack channel, and yeah, just come and see us.
So hi everyone. Okay, let's not. I made a small project in the context of organizing this open science event. So to integrate and improve existing guidelines of organizing hackathons and open science events that are already they're in the cloud. So they are nice, but they require a lot of effort. And the idea is to integrate things that we have used before, that we thought was nice or that we think is helpful to existing guidelines by first listing guidelines. So as, for example, the Brain Hack Global guideline. Collect input in a Google Drive document and then discuss them and maybe also implement them on GitHub repos. So for example, if you already have a template for registration, or you think like this is a question that could be nice and to add it to existing templates, or you have a website template that you think is very easy to use for other people when creating events or even things that I can't think about, then we can add them to a Google Drive document, discuss them, and then decide if we can implement them to existing guidelines. So if you're interested, I will be around the hackathon room, not all the time, but I will be there both days, and just talk to me and we will see. Thank you.
So it's going to be very short because my hackathon was very short. I actually joined the COVIDAS hackathon. I had a short conversation with Stefan, not that Stefan Klein, and I got some very good input from him. So I'm gonna just process his input into the existing decision tree that we already have. But there is a GitHub repo, which only contains README file now, but we're getting there. So if you want to contribute, you can go there, and I've put some links in there that you can use. You can also contact me if you want to contribute. So that was my update.
Hi, you know me already. Now my team has grown to 3 people with different functions. So Chief Financial Officers and Cutlery Master, and Chief Technical Officer and Recycler Enthusiast. So actually, our project was on comparing cortical versus subcortical dynamics, but actually we will not tell you anything about that because you are tired, like it has been a long day, so we'll just entertain you with some memes and some lessons we learned from this hackathon. I hope that's okay for you. So I forced everyone from our board to create a meme, whether they liked it or not. So that's my meme. So we were interested in time series analysis, and so we started with classical statistics, but we kind of got interested in machine learning using weird features in some kind of weird reduced space. Yeah, so that was what we got interested in. You're next.
I did my first pull request today.
Short and sweet. And I was working on trying to plot these metrics we've been calculating between different parts of the brain. And we managed 5 minutes before the end. Plotting things on a surface feels like it's one of those things that there's lots of Python libraries, and everyone you ask is like, oh, yeah, I've seen a library to do that, easy. It did take all day. I had to uninstall a lot of things and start from scratch, but we got there in the end. And you can't see it here, but it is an interactive thing, so hopefully in the future, for the next person who's looking at this, that might be handy for exploring it.
So we are present on GitHub repo, so if you're interested in more like content, you can visit it, or you can join our project next time. Thank you.
Hey. So hi, it is Faruk again. As you know, I have asked 2 questions. Can we explode MRI images, and when it will become boring? Yes, and more research is needed, and I did some research and development. So first things first, now there's a repository on GitHub called Slowest Particle Simulator on Earth. And actually, you can install it by just saying pip install slowest particle simulator on Earth. And you can use it from the command line. You don't even need to script, like from just bash, call it with some nifty file and it will explode it. I mean, some features are lacking, but it is, the core is there. After laying the groundwork, I explored a bit different kinds of explosions or implosions. So this one I call deflating inflation. For those who like to inflate the cortex. So this one I call the brainwashing. Pulsation party. Anyone? Physiology is very important, as we know, right? Yeah, yeah. And this is the favorite, thanks to Johnny for naming it, Benjamin Buttoning. Yeah, and yeah, that's it. Thanks.
So especially Peter and I, but also with contribution from Pim and from other participants over coffee and over beers, etc. We've mainly been discussing a lot. We've been able to extract two specific goals, and the first one was to get the code that Peter had already written for MATLAB in bits, to get that on GitHub, and we've managed to do so. So that's now in the MATBits repository. So this is where a version of an existing toolbox now already exists. So this is cloned. This is basically a MATLAB clone of a specific PyBits version. But we've also been discussing quite a lot. Let's see what I can actually get back. Yes, here. So in discussing like what would be the expectations that people would actually have from such a toolbox, and that's what we've been like documenting here. Like let me scroll up here. So we have we've been looking at existing toolbox packages both in MATLAB and Python. We've been comparing like what are the features. We've been thinking about the scope, like what do people actually want from this, and different people want different things from it. So that's what we've been like discussing, and in the end we've come up with some ideas that we think are important for the architecture, which we've documented here, and it's mainly that we feel that any toolbox, regardless of in what language it would be written, has to first of all start with a representation of all the files that are on disk. But it's not the files that we're interested in; it's the data. And after long discussions, we came up with the idea that the second level representation is in data constellations, or like as like the Great Bear, like a constellation in the sky, or you could also think of it like as data atoms. Like an atom is something that cannot be split. But at the same time, an atom consists of quarks. Like if you bash them together hard enough, well, you can see the details. That's also what we feel with this data, is that it's so there's data and there's metadata, and some really go tightly together. And that's the representational level that we should be looking for, rather than files. We've also been discussing molecules. So what happens if you have multiple atoms, you want to analyze them together. But that's where the scientific research questions start becoming relevant. And that's probably where people will have different research questions. So that's probably the level where a shared toolbox should not go, because that's where the researcher should go in terms of combining the different atoms into a molecule, or even into a more sophisticated representation of where data is linked together. So that's more or less like a very short executive summary of what is written down here. We came up with some examples. We also came up with some exceptions already. So our rules don't totally seem to apply. But I think this is going to be a useful starting point to also extend the discussion to the wider bits and the wider MATLAB community in working together, continue to working together on these tools. Yeah. So that. Oh, yeah. And of course, important is what this. So Peter now has his stuff on GitHub. He did. So he made his first GitHub, actually made his second GitHub repository, but we've shown him doing pull requests to himself, which perhaps is cheating, but we've gone through this whole sequence as well. So I think that's also, Peter also deserves applause for him sharing his code.
The leader of the project, she had to catch a flight. I tried my best to kind of summarize her project. correctly. So I think it's a continuous project from her PhD. The idea is she worked with a lot of patient data. So she's got lots of high quality structural data. She built some pipeline trying to identify lesions automatically by comparing with some control, and she tried to make it a more practical tool for the clinician to use. So the aim of the project is to convert those MATLAB pipeline using like a Python environment. So what we have been doing. So basically, in the current hackathon, it's Barbara and me, but she's got other collaborators. So this project has been continued for a while. You can see here. Yeah, basically there are so many steps. I think we are currently stopped at the second step. That's still a lot of things to go on. I guess for me, what I have learned the most is basically, I think like throughout these 3 days, like people talk about how annoying the dependency can be. So like some software developers on Twitter complain about it as well. I also have a lot of problem with it on the first day. Basically, they write the code, build everything in Mac, and I'm using Windows. So basically, my first day just spent on installing all the correct programs, which at the very end, I was very disappointed because I felt like I couldn't contribute at all. But I learned a lot of things in terms of like this dependency things, and I also managed to install Ubuntu on Windows, so it helps a lot. So at the moment, what I'm trying to do is, so at the very last stage, once they do all those pre-processing, they need to be able to show the lesions on the patient T1 images. So I'm playing around with that at the moment. Yeah, but mainly most of the time I'm just trying to install software or uninstall them. Yeah. Yeah, but I learned a lot in terms of software side. Thank you.
Thank you. So it was a pretty successful hackathon. I'm really happy. I'm just going to give you some numbers just to get an idea. So we had like 10 pull requests, additional pull requests on the main sort of repository, and we had 2.5 first pull requests. Yeah, 0.5, because someone did a pull request and said, well, it's not my first pull request, but it's the first time I do where I actually know what I'm doing. So I really enjoy that. We had 3 new contributors to the project, and also 3 returning contributors, which is always cool. Makes me very happy to see like new faces and old faces, and people are still motivated to help. It's really, really cool. So the main goal was to sort of start working on sort of creating sort of template text that we can reuse to create this sort of like method section. We sort of realized quickly this is like a big thing, so we only started trying to work on the pre-processing part, for example, of a typical fMRI or MRI study. So we've started doing this. So ideally now we have some sort of like building blocks that we can use to sort of see how it would be in practice when you want to use the app to create your like method section automatically. Quite a lot of work was done as well to sort of like going through the documentation, making sure that a new person who just comes to the repository or to the project who wants to sort of know how it functions, how it works, has like all the information or enough information. And also we have this sort of mother of all spreadsheet where all the items of the checklist are kept, and this has been like a lot of information has been added and improved there, so there's been like a lot of work done. And one last number, I think that pretty much 3 boxes of chocolate were eaten during the whole hack track, so I think that was really productive. Good work.
So thank you. My project was mainly about like the organization of open science events and hackathons. I worked on it a bit on the sidelines, like incorporating ideas from this event as well. So I just started like by improving or listing some issues that we already did for BrainHack Global, and started making some issues on the BrainHack Global repository and hoping to incorporate them later. And that was actually mainly it. So thank you.